Well, hello everyone, and welcome back. Today we're going to have the November installment of the IPM Hour. Uh, Today we got Mitzy Porras, who's going to talk about asymmetric species responses to climate change. She's currently a assistant professor, in the biology department at San Francisco State University. She received her PhD from Penn State, working on how virus and temperature affects interspecies interactions among aphids. She received her undergraduate degree in biology and ecology from the Columbia National University. And I took the liberty of looking through her bio, and I noticed several publications on eumestacid grasshoppers, which I had to admit I had to look it up because I didn't know that family. These are known as monkey or matchstick grasshoppers, and they sit with their legs kind of stuck out to the side. So they're kind of strange looking grasshoppers. And she's an author on one of the genera in this family, which I was impressed by. So, Mitzy, go ahead and take it away. Share your slides, and let's hear about asymmetric species responses to climate change.
Good morning, everyone, or good afternoon to everyone. Thanks for that lovely introduction, and it is a privilege for me to share our work today. So today we're going to talk about how the species respond differentially to climate change, specifically to one of the parameters of climate change that are heat waves. So it works. Okay, yes, it works. So climate change is posing a high risk to biodiversity. Heat waves can have profound effects on organism development and behavior, as well as species phenology. The structure and the function of the communities can also be affected by these changes from the low scale, or the low scale, like from the cellular level to the community level. So in this graph, we see here the temperature anomalies for about a century. Here in the y-axis, you can see the temperature anomalies, and the x-axis, the months. So here, color-coded in orange, we can see the temperatures for the last decade, and this black dot represents the highest temperature was recorded in 2019. But well, with the updated graphs, we can see that these temperatures are increasing, and the highest temperature will reach this year. So we have experienced the highest temperature during this century, and we still don't know how the interactions between organisms, let's say solitary bees, pathogens, are being affected by these abrupt changes. So we are going to review some basic concepts and then we'll go to the experiments. So basically, positive and negative interactions among the species are the ones that shape the structure and the function of the species in within their communities. How these species or these interactions respond to climate change is unknown. This map represents the expected changes in fluid, heat stress, water stress in the entire planet. And we are experiencing highest temperature, heat waves for about periods of 2 to 3 days. And these abrupt changes can modify from the cellular response to the behavior affecting the population dynamics, causing cascading effects on the ecosystem functions like pollination. So from literature, from different ecological experiments, we know that microbes can alter the phenotypes of plants and their vectors, as well as other like hosts. There are primary hosts and secondary hosts. So they modify the phenotypes in ways that affect, for example, in most of the most known examples, we have found that viruses would increase the nutritional quality of plants. They also modify the colors, as well as the olfactory cues, making the plants more attractive to the vectors, and this affects the disease dynamics of the entire system. But little is known about how these changes affect the thermal biology, at least the thermal tolerance, how insects are able to tolerate the temperature fluctuations, and the implications at the community, at the population and community level. Those aspects are still unknown. So here we have the extreme heat has the highest incidence in every ecosystem, and although the changes in the mean temperature seem insignificant or irrelevant, these small changes have profound effects at the extreme level or the extreme temperatures. What are going to be the impacts of those changes on these unknown, still unknown interactions? That's something that we should explore. So for some years, we have been exploring these questions, trying to address how the microbes alter the thermal tolerance, the temperature tolerance of insects, how they modify the plant's surface temperature, the nutritional quality. And here we are going to talk about one system. We try to address these questions with this system, that is Osmia cornifrons and Osmia lignaria. There are solitary bees. They are responsible for, in the case of Osmia cornifrons, of a high, they have been used for a long time as a pollinators in fruit crops. And these both species, well, Osmia cornifrons has been introduced from Asia, and Lignaria is a species that is native in North America. Both species have been shown to be susceptible to a pathogen, that is Crithidia mellificae. Another trait about more descriptions about the biology of these species. These species are renewable time. They overwinter as a cocoon, an adult, and they have these interesting nesting, different nesting. So they are solitary. They don't form these the very common hives. So they are known to be very, very efficient in pollination, at least in the case of Osmia cornifrons. So climate change is affecting in different ways the entire system. For example, it is causing changes on the floral availability, the resource availability for bees. It can also modify the phenology or the population growth of the pathogens, and how these changes can modify the use and the impact, in fact, of fungicides. That's something that we have been trying to explore. And we'll basically here, this is the fungicide component, something that is going to be the last part of the conversation, and we are going to talk about how the fungicides may affect the microbial community that is known to be to play a particular role, an important role on the bee health. So talking about a little bit more about these profound changes that extreme heat or heat waves can have on organisms like bees that are known as a cold blood, or maybe they have the same temperature as the environment, so they don't have as complex physiological pathways as, for example, mammals to modify their body temperature. They depend of movement across the environment, try to find the temperature that matches their optima. So these changes affect different organizational levels. For example, to be able to understand the impacts of these variations, we should explore these questions, or try to resolve these questions in a way in which the organisms are interacting, because these traits change usually when the organism, for example, when the organism is healthy or when it is infected. So at cellular level, we can expect a more oxidative stress that will reduce the energetic efficiency. At physiological level, we could expect some species to acclimate and with the time possibly adapt to these abrupt changes. Others will not adapt, and that's the point in which we could potentially lose some ecosystem services, like the efficiency of the pollinators. At the behavioral level, as I mentioned before, most of the insects, they move across the environment trying to find the temperature that matches their optima. And this is a thermoregulatory compensator, thermoregulation, behavioral thermoregulation. It could be also affected by these abrupt changes in the environment. And finally, all these changes may result in reducing survival. We don't know how the ecological interactions, like for example, post-pathogen interaction, the interaction between, an example, Crithidia mellificae and the host Osmia cornifrons or Osmia lignaria are going to respond. And we don't know whether these changes will affect daily and seasonal phenology. All these cascading effects can have profound impacts on pollinations, for example. We also don't know how well. It is likely that the targeted species for fungicides are going to change. The dynamics is likely to change, and how we are going to respond to how these tools like fungicides are going to be used to try to target or match these new dynamics. So here in this experiment, we hypothesize that extreme heat or heat waves reduce the heat tolerance of the host and inhibits the thermal boldness of hosts, as well as reduce the pathogen growth population. By thermal boldness, we mean that thermal boldness is a behavior that is observed in some of the individuals of a population. These individuals are able to move across extreme conditions and expand the distribution of the population. So there are some individuals that are very bold and they will reach other locations and are responsible for the expansion of the dispersion of the individuals, while others are going to remain at the resources like the shelter, the food, but there are some that are explorers and naturally will expand the distribution. So we predict that heat waves will reduce the host and the pathogen performance in ways that could induce mismatch in the host-pathogen interaction. To answer this question and to test this hypothesis, we assess the effects of heat waves on pathogen infection of Crithidia mellificae, and Osmia cornifrons, the combination of these two stressors. And we also measure the pathogen growth rate when the inoculum was exposed to thermal stress. So here I'm gonna review a little bit of concepts of the thermal heterogeneity that was mentioned. So in this picture from Callion, we can see a thermal picture of an apple leaves. So we see high thermal heterogeneous environment where these mites should move across this thermal heterogeneous environment, and they try to match their thermal optima. So here in this example, we have a warm adapted individual and a cold adapted individual move across this hypothetical thermal spectrum. But what happens when the conditions are unfavorable? So probably we'll see changes at the behavioral level. So we are going to observe evasion. They'll probably switch resources. Some of those individuals might be able to have a higher heat tolerance, or it could be also reduced. So here is our question. Does abiotic interaction affect this response? Does heat waves alter the thermal tolerance of the pathogen or the host, or both? So basically, we obtain our cocoons from Washington, and we identify a thermal condition that could be understood as a heat wave for May in Washington and California. That was 30 degrees Celsius. This temperature was recorded by NOAA. I'm sorry, I'm gonna come back here. And part of the population was exposed or was infected with inoculum. We fed some of the insects of the females with 100,000 cells of the pathogen for 6 hours. Then the other part of the population, infected and healthy, were exposed to this heat stress, the heat wave. And we measure a parameter that is called CT max, or the upper temperature limit for locomotion, which means the highest temperature at which a individual stops its movement. So there is a sum of muscular spasm from which we can expect that the organism is going to be more susceptible to risks like predators, or will not be able to survive. We use this experimental device that basically is a water bath that increased the temperature 1 degrees Celsius per minute. We record the temperature, we insert our water bath inside a cabin with a controlled temperature and humidity, and we basically keep observing till we observe the first muscular spasm, and that's the CT mass. So here in this graph, we can see in the y-axis the CT max, and in the x-axis the different conditions, healthy individuals that were exposed to extreme heat or heat wave, the pathogen infection, and the combination of all parameters for corniforms and Lignaria. So we basically observed that corniforms tolerated higher or had a higher CT max compared to Lignaria. Corniforms, we observed that where the average temperature, the average CT max was 42 degrees Celsius and 88 degrees Celsius for Lignaria. Then prior exposure to 1 day of heat waves reduced the heat tolerance of corniforms and Lignaria by an average of 5 degrees Celsius in corniforms and 3 degrees Celsius in Lignaria. The pathogen infection reduced the heat tolerance of both solitary species by 8 degrees Celsius compared to control and 6.5 degrees Celsius. Finally, the combination of both stressors reduced heat tolerance by an average of 10 degrees Celsius in corniforms and 7 degrees Celsius in Lignaria. Then we explored the parameter that I mentioned before, thermal boldness, those individuals, check the number of individuals that were able to take risk and cross these barriers, like thermal barriers that are expected to don't cross because they go beyond the CT max. So here we can see that some individuals, even they could go further the CT max that is expected, that is the upper temperature limit for locomotion. And it could, and using this parameter for in other type of studies would be beneficial because we could also refine the models for distribution of a species. Most of the models use the CT max as the parameter to make the description of and expected distribution of the species, but some individuals can go higher or further. So here this behavioral parameter is tested or is measured by using basically a choice test, where we have a home bottle here in black, where the individuals are initially kept, and then they are allowed to move across this choice test, where we can see that there is a extreme hot sun and an extreme cold. Here in each side of the choice test, we put some pollen as an stimuli for them to move. And we leave the bees over there, the females over there, for 1 hour. We come back and record the number of individuals that make the decision, like cross either cold or warm zones or stay on the home bottle, didn't take the risks. So this figure represents the percentage of individuals of cornifrons, of females of cornifrons, that were able to cross the cold temperature or the cold zone, the hot zone. Here in horizontal lines, you'll see the bars, and in solid color, we'll see the individuals that didn't make any decision or didn't move. So for healthy individuals, we found that 60% of the females were able or risky bold and cross the extreme hot, followed by the staying in the home bottle. But this response changed when we exposed the individuals to the heat wave. So basically, we documented changes in this exposure in both the species. I want to show you the graph later. And there was a significant change, reduction of the individuals that crossed this barrier when they were exposed to the stress by a heat wave. We found even lower, like the pathogen infection reduced even more this number, and the number of individuals that remain in the home bottle were more. Combination of both factors basically reduced by 31% points the number of individuals that were crossing the hot barrier. The individual, the Osmealignaria females, follow a similar trend. Then we investigated whether the thermal stress, the heat wave, and the CT max that was reached by the different conditions when the insects were exposed to the different conditions affected the population growth of the pathogen. And the results here are very interesting. So here we can see in this graph in the y-axis the growth rate of Crithidia mellificae, and here the time. So we gave the shock, the heat shock, let's call it, and then we measure the number of cells that were active on the inoculum. And this is the let's say the population, the growth rate at a constant temperature, that is the optimal temperature for maintaining the inoculum. And it was significantly reduced when we exposed the inoculum to the heat wave. Other temperatures by CT max, the CT max of healthy cornifrons was also affecting the population growth of Crithidia mellificae. In overall, these results suggest that heat waves alter the heat tolerance of both the host and the pathogen. And these shifts may result in physiological and behavioral changes that can modify the pathogen host dynamics. Here we consider that heat waves and infection may drive changes in the thermal tolerance and the behavior of both. And we observed that the heat wave could reduce the tolerance and in both the species. And we can expect that under these conditions, parameters like the body mass or the fat content will be affected, excuse me, will be reduced, and this will have impact on the foraging capacity that solitary bees will have. So these complex physiological mechanics underline significant changes at the individual and population level. Now, I'm going to briefly show some results about how the fungicides may modify parameters that are directly affecting the bee health. And this parameter is how a fungicide may affect the microbial community. Microbial community can provide beneficial impacts to the bees by providing, for example, increasing the immune system of the bees, also producing some toxins that may be defensive against pathogen. So in general, the microbial community, it's an unstable phase that once is impacted by a stressor, let's say a fungicide, could be modified, and we will establish some questions like what are going to be the effects of heat waves on this community, on the microbial community. So we basically took larvae of Osmia cornifrons and fed them with pollen that was treated with mancozeb. We found here, this is a principal component analysis, where we found that the microbial community, the control, was significantly different from the individuals that were exposed to mancozeb or were fed with mancozeb. And here, the richness, the abundance, and the diversity, you can see here, was significantly reduced in the larvae that were fed with pollen treated with mancozeb, with the highest temperature, the field concentration that is recommended. Here, the relative abundance graph suggests that, indicate that the control individuals have a relatively similar community, and this was unstable. It varied when the individuals were exposed to mancozeb. The functions and the impacts of the functions of these genus, we did the analysis at the genus level. That's something that should be explored, should be analyzed to identify what are the genes that are being upregulated or downregulated when these species are being affected, like modified. Here, we know that they are present. We don't know whether they are functional. It's our inference, but at this point we can see that the microbial community, the general microbial community, it is not only the microbial gut. It was a little bit difficult for us to extract the gut from the bees, so we worked with the whole body, and we found that there were differences in the abundance and the diversity of this genus. So what are going to be the ways in which we can explore how multiple complex environments given by climate change affect the phenology of pathogen and and solitary bees. That's something that we should further explore, and how the use of different management tools are going to affect these interactions. So maybe how can we make these applications more effective in a way that they will not put in some risk the health of the solitary bees. As a take-home message, well, despite both the species are relatively phylogenetically close, they display different thermal boldness. It is species specific. The heat waves can reduce the heat tolerance of both species, as well as the pathogen growth rate. The pathogen infection further reduce the heat tolerance of both Usmi species. And the heat wave also exacerbated the adverse effects of the infection, the pathogenic infection on solitary bees' thermal physiology and behavior. Our findings complement prior research showing that climate change can alter host and pathogen systems, and these interactions or these changes may influence the evolution and the maintenance of the species function within the ecosystem. For example, how they are going to perform, their foraging capacity is going to be reduced. That's something that we need to test in the field. So this is basically just rephrasing what I just said. The consequence of heat waves may result in phenological asynchrony of solitary bees and emergence. They also may also modify the pathogen development. So we probably may see that the pathogen will not be able to survive when it is exposed on the surface that it lives. It has a phase that it survives out of the host. Heat tolerance and thermal boldness of solitary bee may limit the pathogen growth, with ultimate consequences on the disease dynamics. But these dynamics depends on the way in which we explore the effects within the interactions, though the organisms by themselves, by separated. What are going to be the implications of such phenological asynchrony of the solitary bee and the pathogen on fungicide application? That's something that should be explored and keep in mind for future research. Finally, I would like to thank our collaborators and funding agencies, as well as the students that were contributing the experimental part. Thank you so much for your interest.
Thank you, Mitzy. That was terrific. Let's see. So for those who have questions, you can either put your questions in the chat, or you can take your mic off of mute. If I will take the host privilege here and go ahead and ask the first question. So what I was thinking about was, and this was part of your last take-home message too. If you look at the CT max of uninfected animals, in this case bees, and you're looking at about 42C, that's basically febrile in mammals, right? Which is a common response to viral and bacterial infection. I don't really know if it's a common response to fungal infection. And my very rudimentary understanding of how mammalian immunology works, I am not somebody who studies mammalian immunology, is that in essence there's a relative benefit of the higher temperature to the cellular and to the immune response, the cellular response, as well as the protein response, and there's a relative detriment to the pathogen. And so what that means, of course, is that you're better at fighting the infection when your body temperature goes up. So I'm curious if at 42 right? They would, in essence, be able to, I would think that temperature would be high enough to be a detriment to the pathogen. But at 38, maybe not so much. And I was wondering if that was true or not. Like, is that dropping of the CT max basically a detriment to the ability of the insect to fight the infection?
That's a very interesting question. Thanks for asking. One of the, well, just trying to address your question directly, is the CT max of a healthy individual. In other systems, like say like the predator beetle, we also observed that when it was infected, the CT max was reduced in some way. Well, we also measure the ATP levels, and those were modified by the infection. So it seems that when the individual is infected, is trying, and we expose them to the CT max experiment, it is not in its full capacity, immunocapacity, it's taking some of the energy. So the individual is not able to tolerate the temperature that is used to tolerate when it's healthy. That's one of the explanations. One of the things that maybe is not involved in your question, one of the interesting things that we observe here in the choice test for the testing the thermal boldness is that usually the behavioral fever was investigated on thermal arenas that expose the individual to a set of high temperatures. And with grasshoppers, they demonstrated that an infected individual with Bovaria, for example, moved to the highest temperature. So in that temperature, the pathogen was negatively affected. So the growth rate was reduced, and it was assumed and it suggested that it's a strategy to control the infection. What is interesting here is that if we offer a variety of temperatures to the individuals, when they are infected, they prefer to remain in the home bottle. So that's something that needs further investigation. It would be interesting to see, for example, what are the genes that are being regulated, that are expressed under these conditions, because basically it challenges that idea of behavioral fever we have. Well, we haven't had the money to explore that, but it was very interesting.
Well, I went full anthropomorphic when I saw that graph. I know when I'm sick, what do I want to do? I want to stay home. I want to stay in bed, and I want to stay home. And when I looked at that graph, I thought, oh yeah, that makes sense. The bees, when they feel crappy, they stay home. But,
exactly.
but I don't know. That's like super anthropomorphic, so I don't know.
It is an interesting assumption, and again, for us, it is striking. We would love to have more funds to explore this, because basically it is unknown. And there is another team, the team that created the thermal arena. They have been working with Drosophila, and they have found similar trends. Thank you for the question.
Hi, Mitzy.
Hello.
Matt's question actually sparked a question in me as well, and your explanation did. So I know basically nothing about immunology, so bear with me here. But I found myself curious when you described the discrepancy in the behavior you observed in the experiment you just described versus the one that you described with grasshoppers seeking a temperature that is deleterious to the pathogen they're infected with. I guess because I come at things from a very applied perspective, it makes me wonder if there would be a way to predict that kind of pathogen host relationship. Like if there is a certain like pattern of coevolution that would make you think like, okay, in this case, a host would probably respond in this way versus in this case, no, the host will not have adapted to, try and deal with the pathogen in this way. And yeah, I just want to hear your thoughts on that.
That's an excellent question, and is a really good inference. It depends on the coevolutionary pathways or trajectories that let's call it like microbes and hosts have had. For example, we explored how a virus, RNA virus that affects plants, grasses, barley yellow dwarf virus, how it modify the CT-max of its vectors. So here we have a different host. This is a secondary host. Both the virus and the vector have co-evolved in a way that the virus benefit the vector in ways that make it more tolerant to heat. In fact, this virus induced the expression of 3 heat shock proteins. There are proteins that basically, let's put it in like colloquial ways, like cover the individual against stress. So it's the first response to stressors, not only temperatures, any type of stressor. And when the aphids were infected, it tolerated 8 degrees Celsius more. It was associated with the expression of these 3 heat shock protein 70. And this expression allowed the aphid to move or to occupy areas of the plants that are warmer. So it expands the niche of the vector. And we see that these both organisms, the virus and the vector, they have been co-evolved for millions of years in a way that the virus will be carried and dispersed by the vector effectively. That's one example, and yes, it totally aligns with your thought.
Thank you. That's so interesting.
Thank you.
Let's see. Do we have any other questions? I don't see any in the chat currently. So I think, Mitzy, that might be it for today. Thank you very much again for a wonderful talk. Your talk will be recorded and will be available through our website as well as YouTube. So we just actually, Emma did the numbers, and we noticed that in terms of the number of times that the recorded IPM hour talks have been watched subsequently was north of, I think it was 2,300 times. So hopefully you will get lots of views on your IPM hour episode. Thanks everyone again for joining today. I just want to remind you that our next IPM hour, the last installment for 2023, will be in December on the 13th. That will be Rebecca Schmidt from the USDA, and she will be talking about biological control in Northwest tree fruit. Steve, is there anything else that I've forgotten?
No, I don't think so. We won't. We don't have a speaker yet for January, so if anyone is interested in presenting, I am looking for a presenter for January, and that's what I know.
Thanks again, everyone. Enjoy your holidays if we don't see you in December.